Okay, we are live. I want to welcome everybody to a really special event. We have Dr. Fazale Rana from Reasons to Believe here. He is a very well-known and predominant PhD in biochemistry. We've been familiar with him from. Oops, I'm getting a live back music. One sec. There we go. We're going to be talking about Human Chromosome 2 and some of his explanations for it. I believe from what I've read on Reasons to Believe, he does accept that is a fusion event, but he asserts that is a divine origin, at least, or as an instance of God actually having some intervention there to make this happen, and we'll get into that. But before we do, I want to welcome our panel. We've got Wayne Fillmore. We have Sam the Scientist, Rational Mind, General Han Solo, Dr. Fiona Robertson, and of course Dr. Rana, we're going to actually refer to as Fuzz from here on out for simplicity's sake by his request. Before we get started, we are going to ask Fuzz a question here that has been plaguing our community for a very long time. It is a very simple question. I basically am asking for the fact that we have so many in our community, as we all know and love, that just doesn't seem to understand the difference between degrade and dissolve. And here's the question, Fuzz. Is it true that degrade and dissolve are synonymous terms when it comes to DNA, and then when DNA is said to dissolve in water, it is actually degrading, i.e. breaking down or up into its constituent parts, and does it lose its nucleotide sequence?
That's not true. There is a big difference between a molecule dissolving and a molecule degrading. So when you add DNA to water, it readily dissolves, and there is an ionization event that takes place because DNA is an acid, and so there's a proton that is released from the phosphate moiety of DNA, but DNA remains largely intact in solution. And in fact, there's a number of different ways one could go about experimentally demonstrating that DNA largely remains intact in solution. Over time, DNA will undergo hydrolysis in water, but it doesn't happen immediately. It'll dissolve and remain dissolved in an intact state for quite some time, long enough to be able to do experimental manipulations in the lab to sequence the DNA and other things like that.
Fiona, do you have a follow-up on that? I think you wanted to ask maybe specifically one of the other things that Ronnie would be saying would be directly related to the hydrolysis of the phosphodiester bond, which actually we had a paper that actually showed that I believe that bond had a half-life of like 10 to the 30th million years or something to that effect. But did you want to ask something, a follow-up on that, Fiona? You're muted, Fiona.
Yeah, I'm sorry, I'm struggling with this blinking machine. Yeah, because when I look at the DNA molecule, and the only way I can see it breaking up and to lose its sequence, the constituent parts, would be that you have to cleave the bond which forms that long backbone, which is the sugar phosphate bond, and it's a phosphodiester bond. And that apparently is the strongest bond within the molecule. In water, as far as I've been able to find out from references, the half-life of that bond in water is 33 million years. And that's because it's a strong covalent bond. But water is a polar solvent. So polar solvent isn't going to touch that strong covalent bond. And that's why DNA can keep its sequence, it can keep its long chain structure when it's in solution. It's that particular bond that causes it to remain as a long chain polymer.
Just to add to that, the sugar in DNA is deoxyribose, which means it lacks a hydroxyl group on the number 2 position of the sugar, in contradistinction to ribonucleic acid, where the sugar is ribose. And that absence of that OH group on the number 2 position is really very important, because it adds additional stability to the DNA backbone. Because in RNA, that hydroxyl group in the 2 position can actually promote hydrolysis of the backbone itself. It's kind of like a self-hydrolysis type of reaction. And this is the reason why you could look at DNA and see it as an elegantly designed molecule that seems to be perfectly put together in such a way to store information inside the cell.
Mhm.
If DNA dissolved when it was in contact with water, it wouldn't survive within the cell nucleus. And then if that's the case, it would be useless as an information storage material. So when you look at the structure of DNA, it's an optimally designed system, in my perspective, for information storage, and then also for being able to replicate that information or to access that information with the cell's machinery during processes like gene expression and so on and so forth.
Does anybody else want to ask anything else on that, or does that kind of sum it up? Does that really kind of put the nail in the coffin on this particular topic that's we've gone over for a year and a half? Sam, you want to add anything to that, or Joe?
Yeah, I just want to, why does so it's optimal for, coding for information of proteins. What does that lead you to conclude exactly?
Why do I think it's optimal?
If you think it's optimal, what does that lead you to conclude?
Oh, if I think that DNA is optimal, what do I conclude from that? I would look at that optimality as evidence for design. I'm, an old Earth creationist / ID proponent, though I wouldn't necessarily subscribe to intelligent design in exactly the same way as the Discovery Institute here in the United States would look at design. But I look at optimization as being one of the features of the cell's chemical systems that points to design. I wouldn't say it's the exclusive case for design, but it's part of a composite case for design. So when I look at the structure of DNA, I see it as remarkably optimal. The phosphate group in the backbone is actually a unique chemical moiety. There's nothing else that you could use to substitute for the phosphate. The two closest possible analogs would be a sulfate group or an arsenate group, but neither one would be suitable. Arsenate doesn't form stable organic esters, and sulfate actually doesn't have the capacity to bear a negative charge once it forms a diester like phosphate. People have shown through studies where they've built analogs to DNA all the remarkable features that the deoxyribose sugar possesses seemingly uniquely. And then with regard to the 4 nucleobases that are found in DNA, they show optimality in terms of UV survivability. They're much more UV tolerant than any other conceivable base that could be used. They also have a set of properties that allow for a distortion of the DNA double helix if there's mismatching in base pairs. And people have shown that if you use other possible nucleobases, you don't get that distortion in the double helix when mismatch occurs, which is critical because it's the distortion of the double helix that actually allows the cell's machinery to recognize that damage and then initiate repair mechanisms. So those would be just off the top of my head, a handful of reasons why I see DNA as, in a sense, not only an optimal system, but arguably you could say maybe a unique system in its data storage capacity. It seems to be ideally put together for its role inside the cell.
Yeah, but I mean, I don't see how you could just infer that it was that was designed that way.
Hang on, Joe. We're gonna get into the intelligent. Yeah, we're gonna get into the intelligent design thing in a few. We're gonna be talking about the human chromosome 2, and I believe that Fuzz, has some kind of intelligent design position on that. So we're gonna get down there in a second. But Sam, did you have a question, a follow up, real quick?
Yeah, and I'm going to have to move to get a little closer to the router just to make sure that everything is all right on my end for reception. But something that actually kind of struck me with what Fuzz said is as a computational scientist, the first thing I would say is that DNA is actually not optimal, right? We have 20 amino acids. You can say, I believe, the number of other alternative amino acids, it's been a while since I did the actual research on it, is something like 22 or 23, right? And we have, in terms of our codons, we have a 3-base system. Whereas, and this is because we have, you have 4 to the third power, you have 64 options for codons. So you have 64 codons for, what is usually endogenously, 20 amino acids. Whereas you could imagine if we had something like what would be the word? A 5-base system, essentially. If we had a 5-base system, then you would only need, 2 bases per codon. And this would actually help with things like translational inefficiencies, the fact that we need multiple copies of tRNA and different isoforms of tRNA. So I would question whether, this optimality, but also under the synthesis of evolution, it seems to be that something being a very quinary, thank you, Steve, it seems to be a necessary consequence of both design and evolution that the most efficient system would be existing. And this is because, of course, maybe if there's a designer, and we can talk about how loose of a hypothesis I actually think that is, you would expect something efficient. But also think about it this way, we would expect the most efficient storage molecule to be used in genetic life if it's evolution, because the metabolic costs, right? If it takes more energy to maintain the genome, well, obviously you're going to be at a selective disadvantage. So I don't know that actually points to design exclusively, because it seems to be a necessary consequence of evolution as well. Like, I would be shocked to see that there was just some vastly more optimal way of storing genetic information in a biochemical context. I would be shocked, and that would really conflict with what I believe about abiogenesis and evolution. And then we can talk about information storage from a computational perspective, but I don't want to get into that because that's highly technical and probably a little left of field.
Yeah, well, and I if I could just briefly follow up, my claims that DNA seems to be an optimal system are not just simply armchair comments that I'm making, but rather are born out of what is in the scientific literature. And there was a famous paper published several decades ago about why phosphate, is essentially critical to the DNA backbone. And so also Stephen Freeland and Lawrence Hurst at the University of Bath in the UK a number of years ago published a series of papers where they showed that the genetic code is an extremely optimal system, maybe even a unique system in terms of its optimality with regard to error minimization. And a team from Israel showed that the genetic code is optimal to harbor multiple overlapping codes. You couple that with work by Francis Crick in the 1960s, his famous paper on the origin of the genetic code, where he basically argues that code evolution, at least large-scale evolution of the code, is simply prohibited. And as a result of that, he argued that the genetic code must be a frozen accident, yet you have Hurst and Freeland coming along showing that the code seems to be exquisitely optimized. And the only way to explain that optimization would be through some kind of evolutionary mechanism. But again, you've kind of got this, self-stultifying problem, if you will. So, this claim that what you're looking at is an optimal system is not, again, a claim that is just off the cuff. And Freeland also has done work recently where he shows that the set of 20 amino acids, the canonical amino acids, are actually an optimal set with regard to the continuous variation of properties with respect to size, charge, and hydrophobicity. And in fact, they've compared the set in nature with random sets that would have been generated. There's probably close to 500 amino acids that would be conceivable that could be used in the cell, and this set of 20 performs, remarkably well. So that optimization is really well established in the scientific literature.
Are you familiar with recent papers that have come out that have actually established that the genetic code is actually context dependent? There's entire clades of organisms where they actually don't have stop codons. It seems to be that right now the consensus is changing, and that it's moving more towards a space where the genetic code, is something that is actually more flexible evolutionarily and is more biochemically contextual. I read the reference actually a couple weeks ago. I believe it was in Trends in Ecology and Evolution. I can look for it if you'd like to see it off air. But I would just have to disagree. I mean, and I did my undergrad and my master's research on this topic, and the genetic code is a lot noisier than I think. I mean, if we took your view, and I'm not meaning this to be anything, but that's sort of ribosome profiling studies, sequence context studies that have now been available with high throughput technology. And it seems to be that rather than being sort of this uniquely crystallized set of events, translation is that it's a very biochemically contextual event. And that's why it's such a vibrant area of research. I could direct you to the work of, for example, Nick Angolia, who does some brilliant work on translation initiation, but I don't want to bore everyone again. Sorry.
Okay, no problem on that. Did you want to follow up on that, Fazale, or do you want to, Fuzz, or do you want to
let it go, because we're
okay
off topic. But
yeah, okay. Let's move on a little bit to human chromosome number 2. I had read on Reason to Believe that, correct me if I'm wrong, you do accept that it is a fusion event, correct?
I think that's a reasonable way to look at it. It does look like it is the fusion of 2 chromosomes. From my perspective, I think when you look at the banding pattern, though, the banding pattern doesn't match identically. It's very similar to chromosome 2A and 2B from chimps. You have the centromeric DNA that's internal, or sorry, the 2 centromeric DNA regions, and then you have the telomeric regions that have seemed to be internal in chromosome 2. It's hard to argue that it's something other than a fusion event. Although I have seen a recent claim by, oh goodness, I'm drawing a blank on the guy's name, but he's with the Institute of Creation Research.
Dr. Jeffrey Tompkins?
Yes, thank you. Who claims that, who questions the idea of fusion, where he's arguing that quote unquote fusion site is actually encodes a gene that's transcribed, I should say, with a transcription binding site. But apart from that, I'm not sure.
I was going to turn this over actually to General real quick. Actually, I'm going to change my mind and put this over to Rational. This is something he's familiar with. A couple of things Dr. Tompkins talks about. He claims that the region is highly expressed, and he also claims, yes, the DDX11L2 gene is somehow functional. We've actually talked about this topic in depth, so people that are listening are kind of, even they are kind of familiar with what we're talking about. Rational, do you want to briefly bring us up to speed on the criticisms we have with Dr. Tompkins' work real quick, and then we'll kind of go into what Fuzz actually thinks the fusion site entails as far from an intelligent design perspective?
Sure. It's been a while since I've gone over this for a few weeks, and I've been filling my head with other stuff with my studies, but I'll try and remember. So yeah, his main point is that there's the DDX11L2 is A, it's transcribed, and B, it's got some kind of crucial function, so it can't possibly have fused and then had the gene sort of appear. It has to have been there all the time, otherwise humans couldn't have survived. So first of all, the transcription level of that gene, or of that sequence, I should say, is very questionable. I mean, it's kind of hard to figure out how much is transcribed, because the actual sort of canonical DDX11L2 sequence is next to it in, let me remember. Yeah, so it's next to it, but the longer transcript of that version, that's right, is through the fusion site. And so a lot of the expression data that you see about this sequence is just about the short transcript, which doesn't include the part of the fusion site, so it's not really relevant. And then I think then, from memory, there's a couple of vague references towards the expression of the bit that goes through the fusion site, but it's very, very low, like 1 or 2 transcripts detected in a massive RNA-seq thing. So I don't think you can argue that it's highly expressed by any means. And there's also been no kind of demonstration of any kind of function. I mean, it's not protein coding as far as we can tell. And it's not, basically Tompkins has demonstrated no kind of function for it, even if it's somehow this one transcript does something.
Actually, so Steve just posted the 3.9 as the value, but actually that is for the transcript that does not, if I remember correctly, that is
Yeah,
the
that's the
transcript
short
that doesn't.
transcript.
The value for the one that does cross the fusion site is something like 0.5 or something. It's not transcribed. At that level, I mean,
Yeah.
you can have random stuff all over the place that can get transcribed at that level. Doesn't mean they do anything. And in fact, there's no evidence that anybody has ever provided that the thing has any functional association with anything else.
Mm-hmm.
It doesn't do anything.
Now, I want to make
One
it
more
clear.
thing
The issue that I would highlight is what Tompkins actually argues for function is not anything. He doesn't, perform any sort of knockdown assay or, an interfering RNA assay or anything. Even though the off-target effects would be crazy from that. The bigger issue is he tries to infer based on co-expression, and the issue is that if you have something that's expressed at that low of a level, the co-expression values turn into a hairball. This is actually, gene function comparison is actually right now my area of research, and I can just tell you, co-expression is always kind of horrifically messy.
Now, Fuzz, I'm sorry.
Which is I just want to highlight, we're not just declaring that, because I know CRISPR and Rational and myself have had this discussion before. We're not just declaring that there are no functional associations.
Can I just add one quick thing before you go, Steve?
Sure.
Just about the binding of transcription factors and RNA polymerase and all this stuff to the region. So I think Tompkins has also argued that because there is these binding of these transcription factors and things like that, and because there's promoters around in that sort of fusion region, that it can't possibly be a fusion. But what Ruhif, shout out to Ruhif, he's done some sort of analysis of other telomeric sequences in human chromosomes, and basically you can see that even in sort of normal human telomeres and the subtelomeres, there is what seem to be promoters, and so basically there's very little difference between a sort of normal telomeric sequence and a promoter sequence in some cases. So
there's just basically transcription binding findings, factor binding sites ubiquitous throughout the genome, basically.
Well, the issue is
kind of, yeah.
And I actually did a rotation on this with looking at peaks in ChIP-seq studies through ENCODE and through replicates in our lab. And I shouldn't say our lab, the lab I was rotating in. And the issue is that it depends heavily first off, what is your scoring criteria? What peak finding algorithm you use for finding a signature in the ChIP-seq, which, by the way, there's no consensus on what's the best, or how to benchmark what would be the best. So we don't even, to that level, it could just be an artifact of the fact that ENCODE has noisy data. I mean, it's almost, as with any public data project, unfortunately, it's unfortunately noisy.
I want to make sure, Fuzz, you had mentioned, there's alternatives out there, Dr. Tompkins stuff. We were just kind of going through it because we've talked about these things before. You're not exactly saying that you're supporting what Dr. Tompkins is saying. You're just saying that it's an possible alternative, correct?
Yeah, I'm not necessarily promoting his perspective one way or the other. And it's interesting to hear your critique of his work. I'm just simply pointing out that kind of in the landscape of ideas regarding, the nature of human chromosome 2, that this idea is out there where Tompkins is challenging the idea of fusion. But I think when you look at the data collectively and when you put all the different features together, it seems like the only reasonable conclusion is that it was some type of fusion event.
And I think everybody on this panel agrees with that.
Yeah, can I just say something? It was what really stood out for me there when you said that was it's when you put all of the different parts of the data together. It's not just one thing. It's all the different parts are coming together, pointing at this single conclusion. Because, and I think because you have so many different pieces of evidence that are pointing to that one conclusion, it would be unreasonable to go off in the general direction of a different conclusion. You'd have to knock down all of those planks, and it would be a remarkable coincidence if all of the evidence was all pointing to this one conclusion, and the conclusion is, completely out of the window, if you see what I mean.
Yeah.
So it's when you said, it's all of the data's pointing that way.
It's a culmination of evidence. It's consilience of all the evidence. I think everybody agrees. Yeah. Wayne, not Wayne. General, real quick. Something on RTP that you and I read last night in regards to these millions of instances. Do you know remember what I'm referring to, General?
Yep. So
yeah.
I have it.
Yeah, you have it. You have it. Okay. Did you want to ask that with Fuzz and kind of get Fuzz's perspective on what the RTP means by these millions of instances of God's interaction, especially with the Human Chromosome 2 fusion site? I mean, did it involve some kind of actual, intervention between a deity, what Dr. Gentry would have called a singularity event? Go ahead. I'm turning this over to General real quick, and then have a little dialogue with Fuzz, if that's okay.
Okay. So, hey, Fuzz. So, yeah, I don't know if you can see this. I've tried to zoom in on the. It's a serving up suite.
I don't think you can see it. Can you read it to him, please?
Okay. Yeah, I'm gonna read it out. Yeah.
Sorry.
Okay. So it says here: is belief in an old Earth the same as belief in evolution? And RTB scholars believe that God miraculously intervened millions, possibly even billions of times throughout the history of the universe, not only to prepare it for life, but also to create each and every new species of life on Earth. Our theistic evolutionist friends would say God initially created and then guided, in some sense, the process through biological evolution to generate life's history. They would therefore take a non-literal view of the creation narrative. So when I came across this, and I also read your 10-year update to Who Was Adam, especially on page 355 and 356, you mentioned the same argument in light of chromosome 2, that because you have here doubts about the cause of fusion, and then you say evidence of genetic engineering, that the Bible's description of animal and human creation suggests a large degree of genetic similarity, which would include similarity of chromosomes, and is to be expected between humans, hominids, and chimpanzees. And then you propose that Human Chromosome 2 offers a hint as to how the creator works. So are you suggesting that this is like creation ex nihilo, billions of times over throughout 3.8 billion years of Earth's history? And I suspect, because me being a Christian, I suspect you're using Psalm 104 in that context in regards to the replenishing nature of the earth. Are these all factors in as to why RTB holds to this particular view? Or maybe if I haven't laid it out correctly, maybe you can clarify any mistakes I've made.
Yeah, and I think it's important. Let me preface my comments by saying that while there's really good broad agreement, reasons to believe among the different people that work as scientists for reasons to believe about the idea that God is intervening throughout the history of life on Earth to accomplish his purposes, there's not exactly a clear consensus, I think, as to exactly what we mean when we say God intervenes in specific situations. There's some range of discussion that takes place. And there's also, I think, a range of perspectives as to when is God intervening, at what level, taxonomic level, is God intervening when he creates, and where does natural process mechanisms play a role. Because we would point out that if you look at, for example, the Genesis 1 and Genesis 2 accounts, there are a variety of different words that are used to describe God's creative activity. Some of those words, it's like bara, seem to imply God originating something new that has never existed before, perhaps even working outside the laws of nature. There's other words like asa, yatsar, bana, that seem to be implying God working with pre-existing material to fabricate something new. There's words like haya and dasha, which look as if God is overseeing processes or even allowing processes to run their course. And so given that, there's a range of ways in which we would argue that God could intervene in the record of nature. And so there's not necessarily a consensus among the team as to exactly what we each would. There's not a consensus perspective in each individual instance. There could be a range of views or even some measure of disagreement. But in a broad sense, we would argue that God is intervening throughout the history of life on Earth, but leave open different ways in which God could bring about that intervention. I don't know.
I'm following that very nicely. That because obviously bara means something new and asar is from something that's already existing. So following from that, and especially trying to connect it with the science, what would be the difference between what the evolutionists would say in regards to the fusion and what RTV says that it's, because I'm sensing RTV is saying that it's a form of bara rather than asar, because asar would basically implicitly mean evolution in a nutshell, but bara would be a non-evolutionary point of view.
Yeah, and I think you could take asar and yatsar and as being God intervening, but not employing an evolutionary process. But I could also see how someone could say those words would be consistent with God employing an evolutionary process. So I'm not so sure that there's a nice, neat, clean way to draw a conclusion from, analysis of what those words mean. And of course, this is also Hebrew, biblical Hebrew, which is an ancient language in which this terminology is, in a sense, being deployed independent of any kind of true scientific knowledge or understanding. And so I think one has to be careful about equating those ideas with, again, modern, scientific concepts.
Right.
But but my understanding, I'm not a Hebrew scholar, but my understanding is asar, yatsar, and bana are roughly interchangeable words. They mean roughly the same thing, and they do describe God's direct personal involvement in a creative activity. It's just that God isn't necessarily originating something new, where Haya and the Shah seem to be more process oriented in terms of how that creation is unfolding, or that process of creation is unfolding. So I would say that chromosome 2 is an asar/yatsar creation, and I think there has to be some divine intervention. But precisely what's happening in mechanistic terms, I don't know that I know the answer to that question.
So,
okay. Are you done with your line of questioning there, or do you want to continue?
Just one more question.
Sure.
I appreciate the clarification, and I'm still thinking that there doesn't seem to be any reason to then doubt the evolutionary model of the fusion in that sense, because I'm thinking because obviously I'm seeing a concordist application, and I guess my question would be now is, well, why do you then reject an evolutionary model, or are you leaving the door open for a possible change of position in the future as time progresses?
When I look at the biblical text, my sense from not only my own reading, but also as I read other scholars in this area, is that God is intervening in a direct and personal way to bring about the origin of humanity, and that the origin of humanity seems to be very difficult to square. Again, in my understanding, and I know that there's people that reach different conclusions looking at the biblical text, is that it looks like, again, there's a personal direct intervention where it doesn't leave room for an evolutionary origin of humanity. And so if one reaches that conviction, then the question then becomes, how can you account for the features that we see when we look at the human genome in light of the genomes of other organisms? How can you interpret that data within a context that's consistent with, some kind of creation / design approach. And so that's one project that I'm undertaking along with a molecular biologist by the name of Anjanette Roberts, who just joined our team here at Reasons to Believe. So we're looking at that question, more seriously. But there's a paper or two on our website written by a theologian named Dan Dyke and a physicist named Hugh Henry, where they propose this idea of God creating hypernaturally. And I don't know that this idea is original to them, but they've done a nice job of exploring the idea and fleshing it out, where they argue that could it be that God is creating by deploying the laws of nature, but God's work as creator is miraculous in the sense that the just right thing is happening at the just right time in the just right way. And so in light of that, when you then go back and you look at questions of origins, it's rather provocative, because even though we're not talking about the origin of life, I think this will help you understand how I'm thinking about human chromosome 2. I'm of the conviction that there's nothing within the laws of chemistry and physics that make the origin of life from a mechanistic standpoint impossible. I think the chemical and physical processes you need for chemical evolution to happen exist within nature. But what it comes out of work in prebiotic chemistry is that the work done in the lab identifying those processes really is critically dependent upon the researcher involvement, the researcher intervention, essentially controlling natural processes in such a way to successfully affect an outcome. And when you translate that chemistry in the lab to the early Earth, you see that it's very hard to envision it being productive. And in a sense, you could argue that organic chemists are functioning in a hypernatural way, that they are affecting the outcome by by creating these low probability circumstances that cause natural processes to do things in productive ways. And you could almost argue that maybe our capacity for genetic engineering is in a sense a hypernatural manipulation of biological systems, where we're exploiting chemical and biochemical processes to bring about genetic changes in hypernatural ways. And so could it be that human chromosome 2 reflects not quote unquote an evolutionary process, but a hypernatural process where a creator is intervening and is essentially causing, telomere fusions, which I just actually did a lit search this morning. In almost every instance where you do see telomere fusions, it's absolutely catastrophic. That creates so much genomic instability that it's catastrophic, for the cell. But so you have, this telomere type fusion event that happens. You now have a mismatch in the number of chromosomes, which either leads to, non-viability, after the process of fertilization, or sterility in most instances. And then you have to have a way for that fusion event to then instantiate itself within the population. And in fact, evolutionary biologists are arguing that chromosome fusion created reproductive barriers that led to the creation of the lineage that ultimately led to modern humans. Well, if it's going to create that kind of reproductive barrier between populations, why wouldn't it create a reproductive barrier within a population? And so when you put all these events together, you're looking at what seem to be a litany of improbable events, which suggests to me the possibility that there's an intervention at play. Exactly what that intervention looks like, I don't know, but you could posit something like hypernaturalism, where you're not necessarily approaching the origin of chromosome 2 in a non-mechanistic sense. You could still have mechanism that you could study if you're a scientist, but it would be in a way that would simply require something beyond what evolutionary mechanisms could achieve without any kind of hypernatural input.
I'd like to ask a quick question before CRISPR goes on to addressing the probability of all these things and how stable the fusion could be and all that stuff. So you're basically saying that because of these series of improbable events, that indicates that perhaps some kind of divine guidance might have been needed in order to make it happen. So my question is sort of in two parts. So the first, I think, is quite an obvious one. Why did God decide to give us fused chromosomes? What was the sort of idea behind that? And the second is, it seems that this argument, you've probably heard this analogy before, but about the sort of shuffling the deck of cards. So you can easily make an outcome that is highly improbable. You can shuffle a deck of cards, you can get a sequence of cards that is astronomically improbable. But does that mean that the act of shuffling itself was, some kind of magical, not magical, supernatural thing, or is it just because we're seeing it after the fact and we're saying, hey, yeah, that happened in the past, it was very improbable, but at the time it's quite a mundane, random thing to happen?
Yeah, and in terms of why would the chromosome appear to be fused? I think I know what you're getting at with that question. I mean, I don't dispute that it does appear to be fused. The question is, did that fusion happen again in an unguided, undirected manner, or was there a divine input involved in that fusion? Which again, I would argue was a divine input.
I'm just asking. Sorry, I should clarify quickly.
Fusion event, and we're presupposing that there's no functional rationale for that, which I'm not completely convinced I'm willing to go down that path, because I've been involved in this now for close to 20 years. And I can remember 20 years ago when the question of, quote unquote, so-called junk DNA came up, there was really nothing of substance that anybody could say about, the existence of junk DNA. And it's astounding to me to see that, week after week, there are papers that are showing function in regions of the genome that people thought at one point in time were non-functional. And just recently, there was a paper published where the point is that even highly repetitive satellite DNA that appears in centromeric regions of chromosomes turns out to actually be functional. That when you start having sequence variation in that highly repetitive DNA, it actually leads to difficulties in terms of spindle formation during mitosis, and may actually be part of the etiology for certain types of cancer. And what's interesting is when the human genome was sequenced, people ignored the highly repetitive regions, partly because it was extremely difficult to sequence, but also there was this bias towards the idea that this probably wasn't functional. And yet other studies are, are identifying kind of as a byproduct of the work, are discovering that areas that we thought were non-functional turn out to be, again, turn out to have function, And so we're immediately assuming that that fusion event had no functional significance, and maybe it actually does, and we just haven't asked the questions, or we haven't made the discoveries that would identify that function. Again, I realize I'm appealing to the future, but given what I'm seeing in the last 15 years, last 10 years, in terms of, again, where the whole junk DNA, project is going, I'm not at this juncture willing to concede that anything in the genome is non-functional.
Okay. Can I just one quick follow-up thing before Sam goes on junk DNA? I'd just like to say, so if you're
just biting his teeth right now because on this junk DNA.
I saw his face when you mentioned junk DNA. So you're suggesting that in the future we'll find some kind of functional reason why the fusion was beneficial, and that's okay, I suppose. But my point for that would just be that one of your points in your sort of series of improbable events of the fusion is the selective sweep. So if there was a functional benefit to the fusion, then that would obviously increase the probability of the selective sweep. Do you agree with that?
I think so, yes.
Mm-hmm.
Yeah.
Okay, I just wanted to clarify that. So I'll let Sam go now if no one else has anything.
There has been somebody who challenged the idea of the selective sweep being necessary. A couple of years ago, sent me a a detailed criticism of why you wouldn't need a selective sweep. And I wasn't completely tracking with the criticism, but that idea of the selective sweep was actually something that Francis Collins mentioned to me in a conversation when we were discussing chromosome 2. And he actually pointed that out as I was going through a litany of all the improbable events, or what I saw to be the improbable events. He added that on to the list. So that's where I got the idea from, was actually Francis Collins in a conversation. But again, to be fair, that point has been criticized. I didn't follow the criticism, but just leave it at that.
Yeah.
So basically, Rasha, you're basically just asking things that have a high probability do occur quite frequently, right? I mean, if you're looking at it from
if you're looking at post hoc, then it's
okay.
But just to add on to that quickly before Sam goes, I'm sorry. So when you say selective sweep, I'm sorry, I'm in the middle of a population genetics class now, so I'm not all on technical all the language. Are you meaning that to say that that it's being selected, or are you just meaning to say that it's sweeping through the population? So, for example, could it be a selective sweep through genetic drift, or is you talking about just selection?
I think it would be selection through that. In other words, that it would be so beneficial that it would take hold within the population very quickly.
Okay.
That would be the argument.
Yeah. Yeah. Okay. So that's what you're expecting to see in the future when we find functionality for the fusion. But because the probability of it being not selected for, of it sweeping through the population just on genetic drift alone is a function of the population size. So I think the estimates are down to, at one point in human history, down to a few thousand individuals. If the fusion happened then I think the probability, you have to remember, if it's neutral, and obviously assuming all the Fisher-Wright model stuff, then the probability of it being fixed in that small population would only be 1 in a few thousand. I say only, but I just have to add that in.
Yeah, I mean, imagine if you had a small enough population, genetic drift could be the mechanism for the instantiation of that feature in the population.
Yeah. Okay. Yeah. Sorry, I'll let Sam go now.
Well, I mean, the first thing I wanted to say is that because I end up having to do just ungodly amounts of statistics, unfortunately, as a computational biologist, I always cringe at the argument of this is really unlikely. And so I don't, because the problem is that likelihood estimates are always, provisional to what your priors are. But more importantly, right, you would never argue something like, if I shuffled a pack of cards and then I, put out the exact order of that deck, it would be 1 over 52 factorial that I end up with that order of the deck, just by, the laws of, by the laws of mathematics, or I should say statistics, or probability. So again, I don't find any problem with what Fuzz is saying. I think I would just say that I'm less convinced by those types of arguments. I have no issue with that. But with the junk DNA thing, I mean, this is something we've talked about, and the issue is I'm assuming that you're referring to the ENCODE analysis that came out a while ago, and there are a lot of methodological flaws with what they called functional stuff. Like, for instance, anything that had transcription was functional, for instance. And we know that introns, which are largely non-functional, are transcribed. So that's roughly, I believe, intronic space in the genome is something roughly like 25%, I want to say. So a large portion of that, right, would actually violate what they say it's 80%. If you remove introns, it's below 80, just by default. Also, there's, significant heterochromatic regions in the genome where the DNA is just so compacted, you're never going to have access from, transcription related or translational machinery getting in there. Just the complexes aren't going to form. It's too tightly packed, of course. But the issue, with determining what the amount of, junk DNA, and we can discuss what that means. I think, it's very context dependent, A, and B, I think that there were some issues with the methodologies that were making those assessments where they were, in statistics, there's a trade-off between something known as specificity and sensitivity. Specificity is your true positive rate, right? So if you have a very high specificity, you can say, if I'm finding a signal, that's probably a true signal. Sensitivity is, or it's also known as recall, is your ability to pick up any signal. So it's going to be your ability, if there's a signal there, you're going to pick up every single signal. But the problem is you're going to have a lot of false positives there. And with the ENCODE project, they were really concerned with, in addition to other methodological issues, they were really concerned with picking up every single possible signal. And the issue is that, like I said earlier, there's no gold standard for picking up those signals. And if you pick up everything, ultimately you're picking up false positives. So we can almost be certain, you can almost be certain in anything with a large sample size that you're going to pick up false positives. So, I mean, we can discuss the particulars there. It's just, I've spoken to Manolis Kellis, and he'll sort of admit as much, which is that with all of these models statistically that they try to use, he directly said in a talk was, I don't really know what this means. I can just predict things based on these signatures. So it's hard to actually know, what exactly is being determined because of the issues that I've outlined there. And then there's issues as what do you define as functional? What are we going to consider a functional region? What are we going to consider a valid human cell line? How many replicates? Technical issues. There's a lot of issues there. So I'm not poo-pooing ENCODE, nor do I want to say that they didn't discover a huge portion of the non-coding genome that does have unexpected roles, probably causally. It's just that we can't conclude based on, I would say that this is one of the rare instances where I'm going to disagree with the journal Nature, and a lot of people have, especially in my field, where I think that there was some impropriety in the statistical analysis there.
Well, and we could have a long discussion about the ENCODE project, because it raises a number of very interesting issues that I think you're touching upon. One of which is, what do you actually mean when you say something is functional? But even if you just set the ENCODE project aside, over the last decade, there have been, discovery after discovery after discovery of regions of the genome that people thought were not functional, that turn out to have, again, functionality, that turn out to be critical in the etiology of diseases and this, that, and the other thing. And so my point would be, given that we're seeing those kinds of discoveries, I'm reticent to conclude that there is no functional basis for why that chromosome 2 is fused. I'm not willing to just simply say it looks like it's a non-functional fusion event that has no consequences, or it may not be important, or may not have a rationale for why it's that way. Just because of things I've seen in the last decade. And so in my mind, I think that the thing that discriminates, let's say, my view from a mainstream evolutionary interpretation of, the origin of human chromosome 2 would be the question of functionality. That if you can truly show that fusion event took place and there's really, quote unquote, no functional consequences for it, then I think an evolutionary interpretation is probably the better way to go. But if you can identify function or some kind of rationale for why that fusion has taken place, then I don't think it necessarily invalidates an evolutionary view, but it does say that one could legitimately look at it from a design/creation perspective. And that would be that would be the point that I would be trying to make.
Fuzz, how would you differentiate between the two, if I can ask real quick?
Yeah, I was actually going to say the exact same thing. I think that the issue is that, right, and this is, something I kind of wanted to bring up with, the efficiency of DNA, but I'm a programmer. I've TA'd multiple programming classes, I've had to grade students' code, and God, it's designed, right? But it's so awfully organized sometimes. And we call this spaghetti code among programmers. Whereas if you were to look at, recent publication that my old lab that I did undergrad and master's in, that they put out, it has my pipeline as part of it. And I'd like to think that's actually pretty efficient, pretty streamlined, if I do say so myself, of course. Quote someone pointing up something about me stroking my own ego. But
oh, that never happens.
Without being facetious, though, right? Is the thing that I would highlight is that also you can have evolutionary algorithms, right? Things where you allow, this is actually what a virus is in many instances, is something that sort of the code mutates and adapts to the environment being the operating system. And so I don't necessarily know that there's any real parameter space in terms of the efficiency of a structure or the functionality of a structure and a designer, because again, like in all of these instances, I can give you 10,000 instances of of software code that's complete. Anyone who's gotten a blue screen of death knows is completely non-functional, but it's certainly designed. So I'm wondering, what are the parameters of what a designer are?
I guess I'm not completely tracking with the question or the point that you're trying to make.
Oh, just what are the parameters of a designer, right? If we want to form a hypothesis or make an inference, we need to know, there needs to be a counterfactual there, right? There needs to be a control.
Okay. I would argue that the designer is the God of the Bible. Is this what you're getting at? And so if that's the case, then I would expect that the designs that we see in nature would reflect an elegance. It doesn't necessarily mean that you wouldn't see suboptimal designs, because suboptimality is part of human designs, where the system is confronted with trade-offs. But I would expect to see elegant designs and expect to see rationale for why things are the way that they are. Is that what you're getting at?
Well, I'm one of the issues that I have with intelligent design as sort of being put forth as a hypothesis is, what's the null hypothesis there? Because like you mentioned, there can be inefficiencies in design, but here's the thing. I'm at a laptop right now. I've got a cell phone right here. If I want to say, is this cell phone designed? Well, I look inside of it. I look at the operating system and I say, oh, okay, that looks like Apple. I go to their factory in China. I go talk to their engineers in Cupertino, California, and I get all of this info so that I can say with a reasonable inference, right? This is designed by Apple. With God, I don't necessarily see there being comparable tests that I can run, right? There's no divine blueprint, so to speak. And there's no, there's nothing where I can say I can investigate into like a history of the design or, what mistakes I would expect, right? If I have a known defect that has, I have a friend who's a electrical engineer who does product engineering. She can tell me, okay, this phone, she works for Samsung, has this known defect, right? There need to be, I've not seen sort of adequately outlined parameters of design that would, remotely compel me in terms of comparable examples, I would say, among humans.
Yeah, I'm sympathetic to the point that you're making, and in my book, The Cell's Design, I attempt to construct a design following in the footsteps of William Paley, who basically drew a comparison between human designs and the designs that you see in biology. And what I do in The Cell's Design is argue that, look, the only known example that we have of an intelligent designer that everybody would agree upon would be human beings. And, and so human beings do certain things with matter and with energy to produce, systems and objects and designs. And so we could, construct what you could call an intelligent design pattern based off of what human beings do. And then if we would argue that humans are made in God's image, which is what the Judeo-Christian scriptures teach, then we would reasonably expect that what we do as designers would in effect be a mimic of what the creator has done. And so that we could in effect use the human designer as, if you will, that standard or that pattern, if you for detecting design. And then you could just simply do pattern recognition type analyses, to determine whether or not the patterns that you see in nature fit the patterns that human designers engage in. So I don't know if that's addressing your question or not, but it seems to me that at least within the construct of science, there is the capability, there are the tools that we would need to A, detect design, and B, to get an idea of the capabilities or the properties of that designer. I mean, the examples that you gave about, concluding that Apple was the producer of a particular phone illustrate that science does have that capability. And I think the question becomes, how can we apply that same kind of reasoning and those same kind of tools to, the larger question of, is there a mind that's behind what we see in living systems? And I think the answer to that question is yes, but I'm sympathetic to your concern in terms of how do we unequivocally establish what that pattern is that we're comparing living systems to.
Hey, I'm sorry, I had some background noise. Yeah, I think the question on the table is basically, what experimental data could we use to determine whether something is more of a teleological or teleonomic. If God did do all this, is there something we can actually do to determine a novel prediction that would point that the only possible explanation, or at least a more probable explanation, would be of a deity or some type of intelligent design? But anyways, Wayne does have a question on this. Wayne, go ahead and go for your question next, please. Wayne, is your mic working? We can't hear you. I think we might be having problems with Wayne's mic. Wayne, you there? Okay, while Wayne's getting that sorted out, can we just get a quick rejoinder on something? Fuzz, you had mentioned something earlier that telomeric to telomeric fusions are catastrophic. Do you remember mentioning something like that?
Yes.
I'm going to turn this over to CRISPR real quick, because we do have some evidence of many cases of telomeric fusions not being catastrophic at all. And I think he just wanted to really quickly touch on that, and then I'm going to turn this over to Joe, because Joe wanted to ask some questions about the intelligent design advocation that you have. So go ahead real quick. CRISPR, could you do a rejoin on that real quick?
Yeah, well, I guess as far as it being catastrophic, I just wanted to check. I mean, are you referring to telomere fusions in somatic cells or germline cells? Either, both, what?
I would say both. Before or this morning, just for fun, I went and did a literature search regarding telomere fusions, and I am not, after doing that lit search, I'm not sure I would necessarily argue that they may be low probability events. I think there's some work done where people are trying to assess what is the frequency in which those kind of fusion events take place. And so I need to spend a little bit more time in the literature, maybe revisiting that particular claim. But everything that I saw, and I was actually looking for instances where people would actually say otherwise. And again, this was a quick literature search this morning, so it wasn't like it was an exhaustive search. Everything I see is that those telomere fusion events basically set up pretty radical genomic instability, and so I have not seen anything that would make me want to withdraw that particular assertion.
Yeah, so that is primarily referring to the idea of breakage fusion bridge cycles.
Yes.
Which is actually almost always between sister chromatids, not different chromosomes. So basically, you lose the telomere on one end of one chromosome. It gets copied, and now both you have both of them right next to each other. No telomere. They fuse to each other. They get pulled apart separately. It breaks in random places, fuses, and you get this cycle going, and that increases your instability. That's actually not it's not really applicable to the kind of fusion you would have in a germline cell, because you're not talking about the cell trying to basically pull these two chromatids apart in opposite directions. That's not what's happening. Now, obviously, you do get a drop in fertility and reproductive ability because it's hard to get the right match of chromosomes. I think it's like 1 in 9 or something like that. But I'm not sure if that really applies to germline.
I'd just like to add that I did email Duncan Baird at the University of Cardiff, who's done a lot of work on telomeric fusions, and I think it's in relation to cancer most of the time. You probably read in your brief literature search, you probably read some of his papers or saw some of his papers. But basically, I emailed him and asked about this, and he said I didn't make it clear if I was talking about the cystic chromatids, as you just mentioned, CRISPR. But when I asked about the sort of probability of this happening in germline cells as relation to his work, he did say that he would anticipate, he's done no research on it, but he would anticipate that the fusions that the kind of fusions that he's looked at would be less, the stable fusions, I should say, the stable fusions would be less common in germline cells. So in germline cells, it would be harder to get a fusion of the kind he was talking about. But again, I don't know if that was cystic chromatids or not. I'd just like to add that.
I've got to step in real quick. Just over the time constraints, Fuzz, I'm appreciating very much so far, this conversation. I know that you probably, have a limited time to dedicate to this, but what kind of time frame do we have for your staying here?
I'm enjoying this thoroughly, and otherwise I would be willing to stay as long as everybody had the stomach to do this. But
we do this for 8 hours at a time, 7 days a week.
I have a meeting that I've got to drive to that's about an hour and a half away, and so I do need to finish up about 3 o'clock, if all possible. But I'm also willing to come back and revisit this topic or any other topic. This has been quite a bit of fun talking with you guys.
What time is that for you? Because I have 3:09 my time. What time is it for you?
Oh, we can't hear you, Fuzz.
What time do you have, Fuzz?
3:10.
Okay, so you're the same time as me, because you said you need to finish it by 3.
Yeah,
sorry,
so
10.
probably need to wrap up.
Okay, let me get, if I can, really quick, 2 people in here really fast. Wayne has a very quick question. He did set this up. And then Joe had another quick question, and I got to let Joe make his question if I can.
Steve, can I just wanted to make a quick comment that adds on to rational so that you don't wait for me and then I have nothing to say. That's related. But I wanted to say, in terms of the genomic rearrangements during cancer, the thing you have to be careful about is that in most cancers that have genomic rearrangements, if not all, they have a TP53 defect, and that gene is known as the guardian of the genome. It's actually the most commonly mutated gene in cancer. So you wouldn't expect cancerous effects in the germline if there's no TP53 mutation. Likewise, you wouldn't expect an endogenous TP53 mutation, because that's going to result in an inviable fetus.
Okay.
Yeah, I don't think he was suggesting.
Well, let's move on. I don't want to get into this technical stuff. We can have a much more in-depth technical discussion with Fuzz another time. Wayne, since you set this up, will you please ask your question, my friend? Now your mic is working.
Can you guys hear me?
Yes.
Yes.
Okay, perfect. Hey, Dr. Ryder, nice to finally meet you.
Yes, same here.
My quick question is so number one, a little bit like a little background. Next, younger of creationist, then I became an older of creationist, and now I'm a theistic evolutionist. And I remember when I was looking at the evidence for evolution and trying to see whether or not if evolution was falsifiable or not. Your podcast, it was one of the podcasts that myself, General, and Steve looked at last night. And it was one where you were on some show, and you were discussing human chromosome 2. And one of the reasons why I love younger creationism is because they had, as you know, a starlight problem. They cannot explain why we see billions of light years of stars if the universe is only 6,000 years old. And so I looked at your podcast, checking that the order position has the same problem, where you guys may not, have a problem with starlight, right? But I would say you guys do have a problem when it comes to this human chromosome fusion, because in any other particular case, we would reckon that the human chromosome fusion definitely is evidence of common descent. If your position is, well, although the evidence says one thing, however, from a theological position, say God did it this way or God did it another way. And so the science is saying, something completely different, but we'll just go with our theology. I watched your video, I became a theistic evolutionist.
Yeah, well, let me respond to this idea that if the science is going one way and the theology is going another way, looking a little bit at the history of biology, and there's a figure in biology that I'm very enamored with, named Sir Richard Owen. And Richard Owen was considered to be the preeminent scientist in the United Kingdom in his day, or one of the preeminent scientists. And many people spoke of him in the same breath as Isaac Newton. He was considered the father of comparative anatomy. He established the modern day museum systems. His accomplishments in paleontology and again in anatomy are legion. And he, prior to Darwin, he was the one who actually developed the concepts of homology and analogy, which we now call convergence. And Owen basically explained shared features through a design/creation framework, where he talked about an archetypical design that existed in the mind of the first cause that then was functionally manifested in the created order. And so what he saw shared features as reflecting common design, not common descent. And in fact, in his framework, he even explained vestigial structures like the whale's pelvis as being necessary because the pelvis allowed the design to conform to the archetype. And he marveled at how amazing it was to have an archetypical design that could simultaneously be modified in a way to produce a wide range of functions that organisms would need to survive in a variety of environments. And Darwin comes along and is very well aware of Owen's ideas and essentially co-opts his archetype and uses that and recasts it as a common ancestor, as opposed to, again, this archetypical design that exists in the mind of a first cause. And Owen's ideas were never, in my view, falsified. They just simply were abandoned in favor of a more mechanistic approach to biology that Darwin offered. Now, I say all this because, and thank you for your patience with that background or that preamble, but I say all that to say that many times what I see is that we look at certain features in biology and we say that those features in biology in effect have only one explanation, namely that they reflect an evolutionary history, they reflect common ancestry, common descent. But as Owen's example illustrates, you can look at the same features from a design framework and make perfect sense of them just as easily as you can make from an evolutionary perspective. And so I would argue that old Earth creationists are not doing the same thing as young Earth creationists, and that we're not ignoring the data, but rather we're saying that the interpretation of the data from an evolutionary perspective is legitimate, yes, indeed, but you could just as legitimately interpret it from a design perspective. But in this day and age, with the way in which science is conducted, a design hypothesis simply doesn't have a place at the table. And so, you know, I would actually say that what we're doing is not the same thing as young Earth creationists. Now, I would argue that where we do, and maybe this is to your point, Wayne, is I would argue that we are allowing our interpretation of Scripture to influence how we think about things, how we look at the world, the types of hypotheses that we would want to advance. And that's what young Earth creationists are doing as well. But what we're doing is trying to look at ways in which we can square our ideas with what is legitimately being discovered in science. So, you know, no way, shape, or form are we discounting the evidence at hand, but really trying to look at, is there a way to accommodate or integrate that evidence within our framework? But I would argue that just because we see shared features among organisms doesn't mean that the only way to look at that is that it reflects an evolutionary history. I would argue that you could, again, make perfect sense of that from a design framework. And that's, in fact, a project that, again, I'm working on, as I mentioned earlier, with Anjanette Roberts, who's a virologist and a molecular biologist. Can we develop, following in the footsteps of Richard Owen, a genomic archetypical model that would be design friendly, that could account for the similarities and differences in genomes of organisms?
You know what? We could probably spend 2 hours just talking about that particular thing you've said in another hangout. I guarantee it. I think, Wayne, did you have a quick follow-up? Because I really would like Joe to ask a quick question if we can, Wayne.
Yeah.
Is that okay?
Yeah, it's fine. My quick follow-up would just be, you know, as far as RTB following the science and everything, I know you guys offer an alternative to evolution, right? So, you know, if you're a Christian, you don't want to accept evolution, you can go to order, which is fine in my opinion, way better than order of creationism. But would you guys ever be willing to reconsider your stance on Adam being the first person? I mean, and I take a concordant view of the Scripture too. I don't think Adam was the first person, but I definitely do think he was the first person to have a spiritual intervention, or he was the first man that God revealed himself to. So would you guys ever be willing to reconsider your stance on Adam and Eve being the first people?
I would argue that we have, over the last several years, visited that and revisited that question over and over again. We've been in conversation behind closed doors with BioLogos, and those issues have come up over and over again. We've listened very carefully to the arguments that they have made, that their biblical scholars and their theologians have made. We've also have looked very carefully at arguments that quote unquote more conservative or more traditional biblical scholars and theologians have made. And at this juncture, come down on the issue as there was, again, a historical Adam and Eve, and that they were the first human beings. But, you know, Wayne, to your point, I mean, there are biblical scholars and theologians who would, you know, interpret Scripture in the way that you are interpreting Scripture. So I think this is an active area of conversation among evangelicals. And so to answer your question, yes, we have considered it, and we've considered it over and over again. And, you know, we have internal discussions quite often about how far could we go on this particular issue. And so, you know, yeah, so we have reconsidered it and have considered it.
Well, I think this is kind of a good place to kind of draw to a close on this. Fuzz, I really wanted to tell you how much I appreciate your time to come in and talk about this stuff. We've obviously could have gone 1,000 different tangents on this. I mean, it's such an open area of discussion, and we've touched on so many different things. Maybe get some more specificity, you know, in a future hangout. I mean, we would love to have you back and engage like this again. So what I'm going to do is, when after I terminate the hangout, you'll get you all the contact information and be able to correspond with you more often. Would that be acceptable?
Sure thing. And I tend to be very slow at answering emails. So if you send emails my way, I will read them, but I may not be able to quickly respond, but I'm happy to interact or communicate. So I'd be happy to set up a phone call or to come back to another hangout. I really enjoyed the time with you guys. Again, I felt like I was on the hot seat, but I'm okay with that. Things to think about.
Sorry about that.
Yeah,
I would.
go ahead,
Can I just say that this has been a fascinating discussion. I mean, I've really enjoyed just basically sitting here listening to everybody, and I have to thank Faz for being willing to do this. So thank you so much.
Yes, very much.
That's
Thank you,
fun.
Wayne, for setting it up. Thank you for my panel, Sam, Rational, General, Fiona, CRISPR. As I said, thank you to all the people watching and listening. You guys have made this community what it is, and definitely have been awesome supporting my channel and stuff. If you've got any questions about anything that we've talked about, email them to me or go to the Great Debate community on Google Plus. You can ask questions there if you join our community, or even just any of us you can send emails to. And I just want to thank everybody, especially Fuzz. That was amazing. Really was informative, and I just can't thank you enough for coming in and talking about it. And with that, I'm going to end the hangout.